The primary 2026 paper examined 309 individuals from 16 Vietnamese archaeological assemblages, including 123 non-adults. It identified 3 young individuals with evidence consistent with congenital treponematosis. One Man Bac case was classified as probable, while another Man Bac individual and the Sun individual were described more cautiously as possible cases. Importantly, the researchers explicitly state that they are not claiming these remains demonstrate syphilis or sexual transmission. At Man Bac, the population-level pattern instead supports a non-venereal form of treponemal disease. That nuance is strengthened in the rewrite. For decades, scientists studying ancient disease have turned to bones and teeth for clues about illnesses that affected people thousands of years ago. Now, prehistoric remains from Vietnam are challenging an assumption that has shaped part of that research. Researchers examining young individuals who lived roughly 3,500 years ago found skeletal and dental changes consistent with congenital treponemal disease, suggesting the infection may have been acquired before birth or very early in life. Congenital cases have often been treated as an important clue for identifying ancient syphilis. But the wider pattern found at these Vietnamese sites does not match the usual epidemiology associated with that disease. Instead, the evidence may fit a non-venereal treponemal illness, possibly something resembling yaws. If that interpretation is correct, some ancient cases once considered strong evidence for syphilis may require a more cautious second look. The research team examined skeletal material from 309 individuals recovered from 16 archaeological sites across Vietnam. Together, the remains represent several thousand years of human history. Among them, 3 young individuals displayed especially important combinations of dental and skeletal changes consistent with congenital treponematosis. Because teeth develop at predictable stages during childhood and often survive well in archaeological settings, abnormalities in their formation can preserve evidence of biological stress long after many other traces have disappeared. 2 individuals came from Man Bac in northern Vietnam, an early agricultural community where previous research had already identified unusually high levels of treponemal disease. The broader age distribution there created an important clue. Many of the individuals showing signs of the condition were young. That pattern fits diseases such as yaws more closely than the epidemiological pattern normally associated with syphilis, raising an intriguing possibility about how treponemal infections behaved in prehistoric Southeast Asia. Syphilis and yaws are classified as different diseases, yet the bacteria responsible for them are extremely closely related members of the Treponema pallidum group. Modern syphilis is primarily transmitted through intimate adult contact and can also pass during pregnancy. Yaws usually spreads through direct skin-to-skin contact and most often affects young people living in warm, humid tropical environments. The problem for archaeologists is that closely related treponemal infections can produce overlapping changes in the skeleton. Thousands of years later, distinguishing one form from another using bones alone can therefore be extremely difficult. That is what makes the Vietnamese evidence unusual. The young individuals show changes compatible with infection beginning very early in life, while the broader population pattern at Man Bac points toward a non-venereal disease. One possibility is that yaws, or an ancient treponemal form resembling it, may sometimes have been transmitted during pregnancy or around birth. If so, modern assumptions about these diseases may not apply neatly to prehistoric populations. Congenital infection has traditionally been considered an important piece of evidence when researchers evaluate possible cases of ancient syphilis. If the bones and teeth of a young individual displayed characteristic developmental changes associated with treponemal disease, congenital syphilis could reasonably enter the diagnosis. The Vietnamese study suggests that this evidence may not identify one particular treponemal disease as confidently as researchers once hoped. That does not mean previous archaeological diagnoses should simply be discarded. Instead, It means that the evidence needs to be interpreted as part of a larger picture. Age distribution, climate, geography, settlement patterns, archaeological context, and population-level evidence may all help distinguish between closely related infections. Whenever preservation and ethical considerations allow it, ancient pathogen DNA could provide another line of evidence. A congenital treponemal case may still represent syphilis, but congenital skeletal changes alone may no longer be enough to make that conclusion certain. This matters because the origins of syphilis have been debated for generations. A major outbreak appeared in Europe near the end of the 15th century, soon after sustained contact began between Europe and the Americas. That timing helped produce what became known as the Columbian hypothesis, the proposal that an ancestral form of the disease reached Europe from the Americas following transatlantic voyages. An alternative interpretation argues that syphilis, or a closely related treponemal infection, may already have been present in Europe before 1492, but was previously overlooked or classified differently. Ancient skeletons became central to this debate. Researchers searched for characteristic combinations of skeletal and dental changes that might reveal where treponemal diseases existed and when they appeared. But that approach depends heavily on being able to distinguish one treponemal disease from another. If several closely related forms were capable of producing congenital changes, some skeletal evidence becomes less specific than researchers once assumed. The Vietnamese findings do not settle the origin debate. Instead, they demonstrate why identifying ancient disease from skeletal evidence can be far more complicated than attaching a modern diagnostic label to an ancient skeleton. Genetic research has transformed the study of ancient infections. Scientists have recovered DNA belonging to Treponema pallidum from archaeological remains in several parts of the world. Those discoveries have revealed a surprisingly complicated evolutionary history involving lineages related to syphilis, yaws, Bigel, and other forms of treponemal disease. Yet ancient DNA is not available everywhere. Tropical environments present a particular challenge. Heat, moisture, and time accelerate the breakdown of genetic material, meaning bacterial DNA can become extremely difficult, or impossible, to recover after thousands of years. Sampling human remains also carries archaeological and ethical responsibilities. Researchers must consider preservation, destructive testing, and consultation with the institutions and communities connected to those collections. For prehistoric Southeast Asia, this means traditional paleopathology remains especially important. Bones and teeth may not reveal the exact genetic identity of an ancient pathogen, but they can still preserve evidence of how disease affected human populations. There is an even larger question behind the Vietnamese discovery. Should scientists expect diseases thousands of years ago to fit neatly into categories defined from modern populations? Today, syphilis, yaws, and Bigel are separated partly by their transmission patterns, geography, and typical clinical presentation. Genetically, however, the bacteria responsible for them are remarkably similar. In prehistoric societies, their behavior may not have matched the patterns scientists observe today. Climate, population density, migration, mobility, and changing settlement systems could all have altered opportunities for transmission. The Vietnamese communities represented in this research lived during a period when agriculture and more permanent settlements were becoming increasingly important across parts of mainland Southeast Asia. Those social transformations brought people into new patterns of contact with one another and with changing environments. The archaeological record cannot reconstruct every detail, but it suggests that ancient treponemal disease may have been more adaptable and less easily divided into modern categories than researchers once assumed. Only 3 young individuals in the study displayed evidence interpreted as congenital treponematosis, and the strength of that diagnosis varies between the cases. That means caution is essential. The researchers are not claiming they discovered 3,500-year-old syphilis in Vietnam. Their conclusion is narrower. These remains contain evidence compatible with congenital treponemal infection, while the broader disease pattern at Man Bac is much more consistent with a non-venereal form. That combination is what makes the discovery important. If future archaeological or genetic evidence supports the interpretation, congenital transmission could no longer be treated as a straightforward fingerprint for one particular treponemal disease. Instead, researchers examining prehistoric remains would need to consider several possible explanations and rely on multiple independent forms of evidence before identifying the infection responsible. The prehistoric remains from Vietnam do not solve the mystery of where syphilis originated. They do something equally valuable. They reveal how complicated the question really is. Closely related infections can leave similar traces in the human body, and evidence once regarded as highly distinctive may have more than one explanation. Archaeology, skeletal biology, genetics, and historical research will all be needed to reconstruct how the treponemal diseases evolved and moved through past populations. Future discoveries may eventually reveal exactly what circulated through prehistoric Southeast Asia and how those infections relate to the diseases recognized today. Until then, these ancient remains offer an important lesson about interpreting the past. Bones and teeth can preserve extraordinary evidence of disease for thousands of years. But identifying exactly what caused those changes requires more than finding a familiar pattern. Sometimes the most important archaeological discovery is not a final answer, but the realization that an old assumption needs to be questioned.